this short video we will be talking about the phylogen, which is a high-density oligonucleotide microarray that is capable of analyzing the bacterial and archaeal composition of complex environmental samples. It contains hundreds of thousands of oligonucleotide probes that are 25 bases long and can target the 16S rRNA gene of bacteria and archaea. A typical experiment includes the isolation of DNA from environmental samples and the amplification of the 16S rRNA gene by PCR with the use of universal primers. Then the amplicons are fragmented and stained with fluorescent dyes. The next step is the hybridization with the probes that are embedded on the surface of the chip. Finally, the chip is scanned and the resulting data is analyzed with the help of specialized software. The phylochip was developed at the Lawrence Berkeley National Lab, and its design was based on 30,000 16S rRNA gene sequences from the Green Genes database. These sequences were aligned and grouped into 9,000 clusters or OTUs with 97 to 100% sequence similarity. The clusters represent 842 subfamilies that belong to 121 prokaryotic orders. Chimeric sequences were also identified and removed from the dataset. The taxonomy of each OTU was based on Burgess' Manual of Systematic Bacteriology, while uncultured organisms were classified according to the taxonomic outline maintained by Philip Hagenholz. The remaining sequences were used to design the probes. Each sequence in a cluster was cut into overlapping 25-mers. The resulting 25-mers and the sequences in the cluster were then submitted to multiple alignments. The 25-mers that were aligned with all the members of a cluster were selected, while the ones that failed to align with even one member of the cluster were discarded. From these subset, the 25-mers that mishybridized with sequences from more than one cluster were also removed. The mishybridizing 25-mers contained a central 17-mer that could be found in sequences from 2 or more different clusters. The remaining 25-mers were reverse complemented into probe orientation. In total, the chip contains 500,000 probes. The probes that target 16S rRNA sequences are called PM probes. Each PM probe is paired with a control probe that is identical except for the 13th base. Positive identification is achieved when 90 to 92% of probe pairs in a specific probe set respond with a signal. Thank you for watching.